Hi everyone. So we have learned so far that a statistical study is a science or applied science of collecting, exploring, and analyzing data to discover underlying patterns, or drawing inferential discoveries um, about data. Um, Today, we will be putting that definition into practice by introducing what the statistical studies life cycle is. And this life cycle is not only pertinent or does not only apply to statistical study, but also to the majority of scientific studies or studies that have scientific methods. Uh, to begin with, the first part of the life cycle starts with um, study with presenting the research question. So again, either you're looking into into conducting a statistical study or any sort of scientific studies, you're more likely to start with presenting the research question. So somewhere at the beginning of your study, you will include a research question or hypothesized actually. A research question could be something as a statement to say, for example, if you would like to, if you decide to conduct a statistical study related to um, a test examining the relationship between unemployment rate and social prosperity or uh, social well-being, then you would say in a research question at the beginning of the life cycle of your study that you are seeking to study the relationship between unemployment rate and social well-being. Or you can actually hypothesize it by saying that uh, uh, there is uh, there is impact or there's good or bad impact of unemployment rate on social prosperity, social well-being. So remember that any study starts with a research question or hypothesis. The second one is the second in the life cycle is developing the study design. It's important to design your study ahead of time because it will designate what the type of analysis and the type of data that you will need. So the study design are categorized into two parts. The first part is when you actually are dealing with data that are already collected. In other words, you're dealing with something that is already available. You don't have to experiment. So observation study are have two parts. The cross-sectional study is involves data that are collected at one single point, and longitudinal study involves data that are collected over a period of time. In the case of studying the um, um, social well-being, relationship between unemployment rate and social well-being, then you're most likely to use cross-sectional study because if, for example, if you were to go out and draw a sample surveys, then you're most likely to collect information at one single point in time. Unless you would like to repeat that survey and go back again and collect survey let's say 3 months down the line, then you're doing a longitudinal study. Um, the second type of study is experimental study, and experimental study is not very applicable and relevant to social science or economic related um, disciplines because it involves assigning treatment and working with control groups. Experimental design is more leans more toward medical science or biological uh, biomedicinal science because it involves again experimenting with your subject humans. For example, if you're to test the effect of a certain type of medication, then you will have a group where you will give them this treatment and then test and then have a control group that you will not give them the medication, and then test the effect of your medication on the treatment group versus the control group. Again, experimental study is not very feasible in social science or economic related studies. So once you develop the study design, you're going to think about you're going to determine the population, and it's important to know what population you're going to target, and depending on your research question and study design, you will designate the population from where the sample will be drawn. So in this case, if we were to, uh, in the case of testing the relationship between unemployment rate and social well-being, our population may be XYZ places, or if we are looking at, if we decide to stratify our population to places that are less affluent, meaning less is- that are in lower socioeconomic class, meaning people who are where locations where less rich people live, then that's where the population group will be. So determining the population could also be if you're not collecting samples, determining the population could also mean determining the data source. Where does your information come from? If the surveys have already been done, so in this case, you could be looking at data points or data sources where those information need to come from. The next one is the determining the variables and sampling method. So the variables and the sampling methods they're important based on so the variables are you decide on the type of variables based on on your study design and based on your research question or hypothesis, and then you decide on the sampling method based on the variable. Some variable require uh, variables decide on what type of samples and data need to be collected. Five sampling techniques that are very common in surveys. The first one is a simple random sample. Let's just not worry too much about the definitions. Uh, Rather, I would like to explain what the objective or of each of these techniques are. In simple random sample, uh, the researcher makes sure that all the samples drawn from a population have equal chance of being selected, that there's no bias while drawing the samples. So for example, if you were to draw five samples out of 20 objects, each of those five samples need to be ensured that they have equal chance of being selected, not one over the other. Stratified and cluster samples, these two types of sampling are closed, kind of close. In cluster sample, you randomly select the entire group and include each group in your sample. However, in the stratified sampling, you select some part of some samples in that group and include them in your sample. So, but however, it's important to know that both of these techniques actually require that the samples are representative of the target population. Systematic sampling, on the other hand, is when the researcher makes sure that the selected members of the survey are selected at a regular interval. So, in other words, let's say if the researcher decides that every second person in the list should be selected for a sample, then he or she will go through the list and select every second person. So a person on the second on the list, fourth on the list, sixth on the list, eight on the list, 10th, 12th, 14th. So all those people will be selected, and that could happen. Let's say if we if the sampling method requires that we select each person entering a door, and we decide to select each fifth person entering that door. So every time, so we will wait until the fifth person enters the door, and we select him as a sample, and every 10th person, and then every 15. So there will be an interval of 5 between each of the samples. And lastly, the convenience sampling sample is, as the name describes, is when you decide to draw your sample from from a place that's very convenient to the to both the objective of the study and also to the methods. For example, if we were to study the the effect of medication on people with diabetes, and then so and instead of going to random places and survey people for diabetes, we go to a diabetic center uh, and um, s- and and establish a sample collection table there at the door of the diabetic center. Convenience sampling is not very common, and it's not highly recommended because it creates a lot of ethical question as well as bias. Because let's say if we were to ask people with uh, if we were to ask people if we were to measure people's happiness, and instead of clustering the geography of our sample into 5 different places where we could pick up samples from random people across all sectors of all state of health. For example, if we go to a diabetic center and we measure happiness there, and that which is a convenient type of sampling, then our result will be highly skewed because almost 99% of the people coming out of that diabetic center um, would um, would provide a result that's biased. And let's say if we ask them how happy they think they are with the economic situations, and they are more likely to say that they're not happy because their state of health actually impacts on how they should answer the question. So I hope the convenience sampling method is uh, it makes sense. Um, After you're done collecting all your samples, you're going to collect the data. So all the samples will need to be collected in a way that can be uh, read or that can be implemented in a lab or in a machine. If it's experimental data, then of course it needs to be taken to a lab. Or if it's statistical related data, then it has to be collected in a way that can be broken down into pieces for analysis, which comes the preparation part. So the preparation part is once you collect the data, you prepare them, you'll make them ready. And in data analysis field, you will learn that data preparation is a very, very important part of data analysis, and a lot of time goes in data preparation. Um, in fact, time there's more time spent in data preparation and data transformation than its use in data analysis. And then lastly, the data is analyzed. So this is where we conduct data analysis and if and use the proper method and model for our data in order to draw analysis. And lastly, once we analyze the data, we report those discoveries in terms of a report or a summary. Or if it's a scientific paper, then there will be sections to report the result of the analysis. Some of the principles of experimental design. By the way, this was it for the for the statistical study life cycle. Some of the principles of experimental design or scientific method I would like to point out is that any any um, any experimental study will need to be randomized and empirical. In other words, they have to be they must not have any bias. This is the nature of science. Science does not accept no subjectivity or bias. It's empirical. It's zeros and ones. The second one is replicable. So whatever you produce in your analysis that you report, that has to be replicable. So if second scientist or a statistician or data analyst gets the data and replicate analysis, he or she will should be able to produce, replicate that analysis on his or her site. Not only the data, but also if, let's say, if a study finds if a study um, if a study find discovers that um, X and X variable has effect on Y and Y variable, so another scientist or statistician should be able to go out and replicate the same discovery. Even he or she has to go and recollect his or her own information. And the other one is verifiability and falsifiability. So any scientific study, any scientific discovery has to be verified. So it has to be falsifiable as well. No knowledge is end knowledge. No science is ultimate. Science is developmental and is evolving. So just because something has been discovered through scientific method does not mean it's ultimate. It has to be verified and also has to be falsified. It has to be able to be verified. It has to have the essence of verifiability and falsifiability. So if somebody verifies it or if somebody falsifies it, then it is okay. It is part of a scientific method. Objective is again when your study must be objective. There should be no involvement of sentiments or subjectivity or bias or feelings, nothing of that sort. It's science. It's zeros and ones. And systematic, that it has to follow a set of methods and techniques to get to a scientific result. Some terminologies that I would like to share is and um, uh, they're not only terminology but also concepts. A single blind experiment is when the subject, and it has to do with experiment, not with data analysis. When the subject or the human the participant who receive a treatment does not know what the treatment is. And the double blind experiment is when neither the subject nor the researcher scientist know what the treatment is. So if a medication is given to a participant, if the participant does not know what the medication is, so that he or she does not bring any bias and let the effects develop without any bias, then it'll be single blinded. And if even the doctor, the person who gives the experimenter, does not know what that medication is, if both don't know, it's double blind experiment. Treatment is again a condition that you apply to a subject, an experiment, which in this case is a medication or anything that you apply. Subjects, depending on your study subject. If it's humans, we call them participants. Otherwise, if it's objects, we call them subject. But subject is the overall term. Dependent variable. Independent variable is explanatory variable. I'll have a lot more videos about this when I get to advanced data analysis. But independent variable is the variable that explains the dependent variable. And then dependent variable is the response variable that actually shows the effect of an independent variable. In other words, giving Medicaid or independent variable in the case of unemployment rate will be unemployment rate will be the independent variable, and the outcome of social well-being will be the dependent variable. Control group, treatment group, these two have applicability with experiments. Control group is the group that is not given the treatment. For example, if the experiment involves giving medicine to a group of two groups, one of them is control group, another one is treatment group. Control group will receive the placebo or is not or not giving anything at all, but the treatment group is given experiment to test the difference of the effect. Confounding variable is, as we have talked about it on the video on correlationships, that these are the variables that actually that are underlying variables. So let's say if we're looking at two variables and how they relate to each other in the case of unemployment rate and social well-being, then we say no, actually unemployment rate is has impact on social well-being, but there are other things, confounding variables that also plays a part. For example, when you're unemployed, then you're more likely to let's say uh, become depressed. And when you become depressed, then you become unhealthy. And when you become unhealthy, so those all those things, all those variables, all those conditions that are around a variable that are underlying uh, variables that we don't test for them, and we do sometimes test for them in a control variable, and and you will see how we factor in confounding variables in advanced data analysis models. But just to understand that confounding variables are any underlying or secondary variables are that are missing in testing the relationship between two variables. But just because they're missing does not mean that we don't know them. Uh, Placebo again is any substance or any treatment that looks like the actual treatment but is not the actual treatment. For example, if we have a medicine, then we have a medicine that looks like medicine that we give it to a participant or a patient just to see the effects, but actually is a placebo. It's just a fake substance. And placebo effect is when when a participant or a patient does not have the actual treatment but have the placebo, and we would like to compare the effect of actual treatment versus the placebo effect. So treatment versus placebo effect. The treatment is when you actually have the actual medication, and placebo is when you actually have the fake medication in the case of experiments. Informed consent, this is very important also in experimental studies that any person that participates in your study have to provide their consent that they are willing um, to participate in your study, and you also and the researcher or experimental is also responsible to make sure that he or she discloses everything to that participant about what the goal of the study is and what are procedures and process involved in the study. And data ethics is pretty much since in data analysis we don't we if we have the data already available, if we are extracting it, pulling it from a data source, since we're not dealing with the participant, we have to take into consideration data ethics and data integrity. So depending on the industry where you work in, there are a series of protocols and ethics and regulations that an analyst or researcher needs to follow in order to stay compliant with the uh, with the ethical procedures and agreements. Um, this was it, I guess, for today. I hope this video was helpful. I know it was a little bit long, but these are very, very important parts of a scientific study or statistical studies life cycle. I hope it was helpful, and thank you so much for your time. Have a great day.